So when we consider the life cycle of the malaria, the infective stage is the parasites which are present in the salivary gland of the vector. So in the case of Sri Lanka, the vector is the female Anopheles culicifacis mosquito, right? So when the female mosquito they take the blood meal, the sporozoites transfer into the human body. So this is taken by the macrophage and it is lodged into the liver cell, hepatic cell. So inside the hepatic cell it will multiply. So in here sporozoites normally spindle shapes. After enter into the liver cell it become round shape. Here it will multiply a large number, right, more than 10,000, and accordingly the species the number may vary. So after rupturing these hepatocytes, these merocytes release to the bloodstream, and in bloodstream they will infect the red cell. So according to the species, again the red cell, the maturity of the red cell may vary. For example, in the case of Plasmodium vivax, they infect the young RBC, but in the case of falciparum they can infect all RBC, old one or the younger. But in the case of malaria, older one. But in the case of ovale, younger RBC, they will prefer to infect. Right after infection, inside the RBC, there are some morphological changes. So these round shape merocytes enter into the red cell. It become a ring stage. Then it become a amoeboid form. Then it become a schizoid form. This schizoid, again the number of schizoid may vary according to the species, but here 8 to 32 the number, right, it may vary, right, but in these schizoid merocytes in liver cycle more than 10,000, but in erythrocytic cycle the merocytes are normally 8 to 32. It's also depending on the species. Then after fully matured this schizoid, the red cell will rupture, and this schizoid, inside the schizoid we can see the merocyte. This merocyte releases into the bloodstream and it will infect the red cell and it become a gametocytes, right. Then the vector, again when they take the blood meal, these gametocytes enter into the vector and there are some modification inside the vector and it is fused together, form oocyst, then zygote, kinetocyst, and it goes to the develop into the sporocytes, right. This is the life cycle of the malaria, right. What is this and what is the infective stage? This is the Balanterium coli tropozoite, because normally Balanterium coli is the only one protozoan ciliate, right. It is a very larger one protozoa, and here how we differentiate the this one cyst and tropozoite. Here tropozoite, here outside you can see the cilia. Here this is the cilia, you can see outside. This is the locomotory organ. So by using that they can move. But in the case of cyst, not like that. This is elongated, little bit oval shape. But in the case of cyst, this is round shape, and outside, very outside, we can see the that means the cyst wall, right. And here you can see the kidney shaped nucleus. This is the kidney shaped nucleus, Then here you can see the contractile vacuole 1 or 2. Here the one, right? You can see the 2, right? Here you can see the, that means the nuclei. Here you can see 2 nuclei. One is the kidney shape, other one is spherical shape. So this is the tropocyte of the Balanterium coli. Infective stage is the cyst of very mature cyst. Right here, name A and B. A is the cyst of Giardia lamblia. B is the tropocyte of the Giardia lamblia. So how to differentiate these two? This one, the first one, cyst is oval shape. This one, tropocyte is the pear shape. Right here you can see very large nuclei. Here you can see, here carefully when you are observing, here you can see the sucking dish, right? And here you can see the flagella, right? And here you can see undulate, this one, the exostyle, right? Here you can see this one, remnant of the flagellum, right here. Then this is exostyle, that center. Then this is the nucleus, and you can see here two layer, two cyst wall, thin cyst wall, alright. So these are the features you have to differentiate. Then the infective stage is the cyst, mature cyst. This is we call giardiasis. What is this? This is the Trichomonas vaginalis tropocyte. So mode of transmission, sexual contaminant intercourse. So here how to differentiate from other species, especially the flagellate. Here you can see only one nucleus. This is the nucleus, right, dark purple color. Here you can see exostyle, right, and here you can see flagella, 4 to 5 flagella. Here in this area you can see undulating membrane. By using this undulating membrane you can differentiate the Trichomonas vaginalis and the Trichomonas hominis. In the case of Trichomonas hominis, right, the undulating membrane full of the body you can see, but here half of the body in the case of Trichomonas vaginalis. That main features, then one nucleus very larger and undulating membrane. Then name the parasite. What is the infective stage? This is actually Iodamoeba botulism cyst. The infective stage also cyst. How we can define it? It is Iodamoeba botulism mature cyst. Here you can see single nucleus. This is actually iodine smear, right? Single nucleus, right? Single nucleus, right? Here you can see this one. Here you can see dark color. We will take, right? Here you can see the glycogen mass. In iodine smear normally it is brownish color. Here you can see the single nucleus, right? What is this? This is actually Rhizospora belli mature oocyst. Here we call oocyst altogether. This is sporocyst. We call sporocyst. Right, inside the sporocyst you can see here. So here are two. You can see the two sporocyst. Oocyst contain two sporocysts. This is sporocyst contain 4 sporozoid. So totally it contains 8, right? That means here 4, other one 4, right? What is this? What is the infective stage? So here we already discussed this also, the gravity proglotis of Taenia solium. Here you can see this is 6 centimeter, 6 millimeter, sorry, this is 6 millimeter, and this one 12 centimeter, sorry, 12 millimeter, this is 12 millimeter, other thing, the normally it pass out with the chain 3 to 4 segments together, and here you can see lateral branches both side. Here you can see the lateral branches. It should be 5 to, actually 7 to, that's 7 to 13, that means less than 15. If you identify that, this is Taenia solium. Other one, the infective stage is the Cysticercosis cellulosea and the egg also infective stage in Taenia solium, right? If Cysticercosis cellulosea larva infected, it become a adult and it produce the teniasis disease. But if egg is infective stages, this egg become a larva Cysticercosis cellulosea. It produce cysticercosis. This is called cysticercosis. This is very dangerous because it affect the nerve system, and so it produce neurocysticercosis. So it is very dangerous. It lead to death. Right. Then what is this? Give the reason. So this is scolex of Taenia solium, and reason 2 rows of hooklets and rostellum. So here this is the sucker, right? This is the sucker. This is common for Taenia solium, Taenia saginata. This is the 2 rows of hooklets. This is the rostellum, right? Here you can see globular shape rostellum, right? But in the case of Taenia saginata, you cannot see the 2 rows of hooklets or rostellum. Only you can see the 4 suckers. Another thing, this is the Taenia scolex of Taenia solium, globular shape, but in the case of saginata, the scolex is pear shape, right? Then this is the coccidian parasite. This stage found in the feces. Name the parasite. Name the strain used to identify this. Actually, name the parasite. This is Cryptosporidium parva mature oocyst. Normally, by using saline and iodine wet mount, we cannot identify the Cryptosporidium parva oocyst because the size is smaller. Other one is, it is very acid fast stain. So normal stain, by using normal stain we cannot identify. So we have to use the special stain. Normally for the acid fast parasites, the organism, we use modified cell lesion. Actually you had read the modified, then the word modified cell lesion staining is the staining technique used to identify the Cryptosporidium parvum. So here the spelling of the cell lesion should be, remember, if you not write correctly you will lose your marks. So this is the oocyst. This is the oocyst. Inside the oocyst you can see the sporozoites, right? 4 sporozoid is present. 4, right? 4 sporozoid is present. Right. Then what is this? This is Cercaria of the Cystosoma species. So this is the infective stages of the Cystosoma species. According to the egg morphology, we can differentiate the species first, but the Cercaria and Miracidia somewhat difficult to identify. But how to identify this area? You have to remember fork tail, and this one the head part. This is the body, right? So what is this? This is actually hydrated sand of the Echinococcus granulosus. So when we consider the Echinococcus granulosus, this is definitely host is the dog, in accidental host is the human. So the infective stage is the egg. When we take the egg, when we ingest the egg, it lodge into the liver cell. It become a larval stage. We call hydrated cyst. The larval stage of the Echinococcus granulosus we call hydrated cyst. So hydrated cyst is filled by the fluid, like liquid things, fluid. When we take the drop of fluid and when we observing the fluid, we call hydrated sand, right? So hydrated sand contain trapped inside. There are so many brood capsule or daughter hydrated cyst. Sometimes if it is fully measured, it will fractured and internal parts will present in that liquid part. So when we take drop of liquid part and when we observe, we can see this brood capsules and these things, proglatids, we can see. Name the parasite that show the life cycle. Name the culture medium used here. This is the life cycle of Entamoeba histotica, right? Because this part extra intestinal amoebiasis only you can observe in the Entamoeba histotica. So we have to differentiate. Only the Entamoeba histotica has ability to invade the wall of the colon, and it will multiply, then lodge into different organ and lodge there, and it will multiply. So in the case of intestinal environment, and so intestinal amoebiasis, we can see both in acute condition cyst and tropocyte, but in the extra intestinal amoebiasis you can see only tropocyte, not seen the cyst form. Right, here we can use culture media, diamond media, Robinson media. Right, here in which parasite you can observe in these stages, give the possible reason for your identification. So here you can see this is the ingested RBC actually. This is the nucleus, right? And by using this we can confirm Entamoeba histotica. Sometimes you may think it may be Entamoeba dispar, but in the case of dispar, when we consider the tropozoid, Entamoeba histotica contain ingested RBC. But in the case of Entamoeba dispar, which not contain red cell, ingested RBC. So by using that we can differentiate the tropozoid. But in the case of cyst stages, this is the cyst. This is the cyst stage. But by using cyst stages, we cannot differentiate Entamoeba histotica or Entamoeba dispar. If we want to differentiate, we have to go for the molecular study, or else isoenzyme studies we have to go, right? what is this name AB, right? This is the actually the adult cystosoma species. A is the female, B is the male, right? What is this and what is the definitive host and the intermediate host? This is the Echinococcus granulosus adult. Right here you can see only three parts, actually four parts normally. This is head part, immature cyst, mature segment and gravid segment, right? Actually altogether the size is 5 millimeter or 0.5 millimeter, very smaller one, right? 0.5 millimeter, right? 0.5 millimeter, right? But when we consider the head part, it's similar to the tinea solium, and the gravid segment olea similar to tinea solium, but the size is very smaller, right? And here only one segment you can see, but lateral branches, number of lateral branches you can see similar, right? Definitive host is a dog, right? What is this? What is the use of this? This is thin and thick blood film. Normally this is used to identify the blood parasite, especially thin blood film. We are used to identify the species of the plasmodium, right? Right, name ABC. A is the Ascaris lumbricoids, fertile, decorticated egg. Outside you cannot see here. If it is corticated, you can see outside dark brownish color, irregular shape, right? Here not like that, so we cannot say, right? Then B is the Trichurus trichura X. This also immature one. C is the hookworm egg. Infective stage is the, in the case of A, mature embryonated egg. B also mature embryonated egg. C the floriform larva. Now sometimes A and B you can say larva containing egg. Right, A, B, C, D. A is the Menzoni egg. B is the hematobian egg. C is the Japonicum. Here you can see the lateral nub. This is the terminal nub. Here you can see the actually lateral terminal spin, here lateral spin, here knob, right? And this one cercaria of the cystosoma. Name the parasite that show the life cycle. So here by using this you can clearly identify, by using these features you can say this one. Other one is you can use by these two features you can say Ruchiradia bancrofti. The vector is the female Anopheles culicifacius mosquito in Sri Lanka. Okay, thank you.